Okay, so before we get started with the talk, I just want to introduce our speakers. So Rick Silverman is professor of medicinal chemistry at Northwestern University. He's author of several books that are used worldwide and is most famously the inventor of the drug Lyrica. Hande is a professor at the Feinberg School of Medicine and is a world-leading researcher in upper motor neuron biology and specifically how it applies to the disease pathology of ALS. They're going to give a joint seminar today. But before we get started with that, I wanted to ask you guys a couple simple questions. Like first, Rick, besides Lyrica, what is your favorite drug and why?
So it's besides Lyrica. Okay. No, I think I really like these checkpoint inhibitors that are using our immune system. You know, theoretically, they could work for everybody, but they only work for a small percentage. And so it really points out how important the immune system is and how different it is from patient to patient, and why we really have to have personalized medicine and gear our drugs specifically for certain classes of problems and people. And that's something I hope to talk about today as well.
Yeah, fantastic. Yeah, I love checkpoint inhibitors as well. It's incredible that Jimmy Carter is still alive. I mean, yeah. So, yeah. How about Hande? How about you?
My favorite drugs.
Mm-hmm.
A couple, actually. One of them is growth factors, because now we realize that we can actually use growth factors as drugs. So that, to me, that's very important, especially as promoting the health, the survival, differentiation of diseased neurons. So that's one of my favorites. And the other one is these DUBs, you know, they add a ubiquitin, they remove a ubiquitin. So if we find a way that we can modulate them in the most effective way, we can have numerous controls over, you know, protein aggregation and cellular function. So the drugs that specifically modulate, you know, a distinct aspect of one DUB, you know, would actually be very useful in clinic. So those are my favorites.
Yeah, great choices. Yeah, so we have a lot of you know, drug hunters or aspiring drug hunters in the audience. You know, Rick and Hande, both of you, if you could go back in time to when you first started your career in drug discovery, what advice would you give yourself?
Yeah, well, unfortunately, early in my career, I really wasn't focused on discovering drugs. I was just doing academic science and fell into this when I realized that we can use this basic science in developing drugs. And so if I had a little more energy maybe and enthusiasm for this, I would have decided to start a company earlier to manage those discoveries, because I've had compounds licensed to companies that have gone, you know, to a certain distance to show that they're pretty good, but then the company runs out of money, changes direction, and those drugs could have really helped people, and they're sitting on a shelf.
Yeah, definitely. Hande, how about you?
So to be honest, I'm not I don't make drugs, or I'm not a medicinal chemist. So I have not made any drugs. So our collaboration with Dr. Silverman is valuable because he's actually the medicinal chemist, and we're trying to investigate whether the compounds that he identified and characterized have any utility in, let's say, improving the health of diseased neurons or within the context of a disease. So I'm not a chemist myself, and I have not made any drugs. And I think that requires a very different set of expertise, and I don't have it, unfortunately.
No, it's all right. Yeah. Well, thank you both for, again, being here. So Rick and Hande are going to tell us more about the science behind some of the new programs going on in their labs. One of which, I guess, is leading to a new startup company called Akava, which is Hebrew for inhibitor or inhibition. So I'll let you take it away.
All right, let me share my screen. Okay, you can see that, Dennis?
Yes.
Anybody? Good. Okay, so the basis of all this is protein aggregation, and I want to tell you about the development of a drug. Came from my laboratory. We originally called NU9, and it's now referred to as AKV9, coming from our company, Akava. And the focus is upper motor neuron diseases. So it's not moving. Oh, there you go. All right, so let me tell you briefly about this company that I started. As I said, because I had had some unfortunate problems with other molecules that we had initially worked with. And so just an overview of what we're going to be talking about today, that all of the science we'll be talking about really came from my laboratory. And of course, all of that really comes from my students and postdocs and even some undergrads had a lot to do with it. I have outstanding collaborators. Hande is one of them. And so we have a pipeline of compounds that we're going to be in licensing. Right now, our lead molecule is this AKV9. We're going to target ALS as the treatment. We have very good preclinical data, efficacy and safety data, and recently it got orphan drug designation. It really has at least 5 different mechanisms of action, all focused on upper motor neurons. And so this is really going to differentiate this molecule from others in the field because of the multiple mechanisms and problems that can cause disease. Also, because of these different mechanisms, there are going to be multiple indications that will be important, including, say, ALS. They're all going to be related to upper motor neurons. Primary lateral sclerosis, hereditary spastic paraplegia, Alzheimer's. These are all problems that can be caused by the upper motor neurons. We've shown already now, and you'll see that the AKV9 works better actually with the two ALS drugs that are out there. So drug combination really will be beneficial. We're writing the IND as we speak, and hopefully that will be submitted very early next year. And this is all being helped by our world-class scientific advisory board. They all are very experienced in ALS in all phases of development, including research in biomarkers. So Akava then is an early stage biotech company. I founded it a couple of years ago for first-in-class small molecules for unmet medical needs, focusing in neurodegeneration and oncology. And it's called Akava because Akava being Hebrew for inhibition. All the projects are related to some kind of inhibition of protein aggregation, such as AKV9, inhibition of enzymes, inhibition of cancer. And so we call these inhibition therapeutics. AKV9, say, is a protein aggregation inhibitor that has been shown to, we think, the first compound to improve the health of upper motor neurons that then would lead to motor neuron diseases. And so eventually we hope to in-license other molecules from my laboratory and expand the pipeline. So ALS is a horrendous disease, fatal neurodegenerative disease. It affects the motor neurons, not only in the brain, the cerebral cortex, but down through to the spinal cord, the lower motor neurons as well. There's no cure, not even an effective therapy for ALS as of now. About 2 in 100,000 people are diagnosed with this disease, but now with improved diagnoses, this is becoming larger. The disease leads to the eventual death of the motor neurons and then the death of the patient in a really short period of time, usually 3 to 5 years from symptom onset. Most of the cases are sporadic. There's really no clear etiology. Only 5 or 10% are familial. And so AKV9 then is going to be an important molecule, we think, for this problem. Motor symptoms result in the degeneration not only of the upper motor neurons, but also the lower motor neurons. But the upper motor neurons seem to display early degeneration, and we're hoping that by maintaining the health of the upper motor neurons, they then will be able to continue to communicate to the lower motor neurons. And so this molecule shown here, AKV9, I think is the first compound to improve the health of these upper motor neurons. Well, it's been a sad story with regard to translation up to this point. All the ALS trials fail, and they're based on mouse models that don't seem to translate to humans. We've found that upper motor neuron degeneration occurs very early, and so we're looking at the cellular roots of neurodegeneration that are common between upper motor neurons of humans and upper motor neurons of mouse models. And so we're hoping then by having an effect on the cellular roots of neurodegeneration that are common, that this should translate to humans as well. ALS is really not just a single disease. It's complex, heterogeneous from patient to patient. You can have different roots of cellular degeneration. And so having one molecule for one particular mechanism is not going to serve the community well. But having a drug that can address several roots that would be important to neurodegeneration would have an effect on a larger population. And as you'll see shortly, AKV9 acts on at least 5 common roots for cellular degeneration, and it's synergistic with approved methods. So if I can compare what used to be done to what we're doing in Aqaba, as I said, previously there was no translation. The mouse models, you can have an effective treatment in a mouse model and it just doesn't show up in humans. But we're looking at the cellular cause and mechanism focused discovery. And so we think that since those are common, that should have an improvement. Previously, upper motor neurons really weren't considered as a strategy for drug development, but we think that this is really an important type of neuron that we can maintain its health, and that will lead to an effective treatment. Previously, cells that weren't even related to motor neurons had been used to screen molecules, but as I say, we are focused directly on upper motor neurons, which we know are important for this disease. and previously no translation. But now it seems that the species level is not going to be the place for translation. It really has to be a cellular level. So there are a lot of differences between humans and mice at the species level, but upper motor neurons really are very similar in the two species. And the causes for neurodegeneration down here are the same causes for neurodegeneration in mice as well in humans. So let's focus on these. The mechanisms include protein aggregation. AKV9 inhibits protein aggregation. And this is the aggregation that causes the disease upper motor neurons. AKV9 improves mitochondrial stability, restores the integrity of the inner membrane. It also improves stability and integrity of the endoplasmic reticulum, maintains the cytoarchitectural integrity, apical dendrite stability, and stops the upper motor neuron degeneration in the motor cortex. And the axons are really important since they have to reach from the brain down to the spinal cord, and AKV9 improves axon outgrowth, branching, and arborization. So we're now going to take a look at each of those five and actually look at some data that we have with them. First of all, let me again say that protein aggregation is really a common problem in all neurodegenerative diseases. And so if we can have an effect on protein aggregation in ALS, we're hoping that that will have an effect in other neurodegenerative diseases, and we already have some indication that that's the case. Well, let me tell you where NU9 came from. So it started out actually in a high throughput screen, not this molecule, but we ran a high throughput screen, 50,000 member, to look for molecules that inhibit protein aggregation caused by mutation in the gene that causes us about 25% of the familial ALS. This is the superoxide dismutase 1 gene. And so we overexpressed this mutation, PC12 cells, and then set up two different assays to look for molecules that inhibit the protein aggregation and the cytotoxicity resulting from that aggregation. And we came up with three classes of molecules. We then spent several years optimizing these three classes. And when we had done that, we found that this molecule, NU9, seemed to have the best properties with regard to pharmacokinetics as well as toxicity and efficacy. And so that's the one that we're moving ahead. And on the right here, I show you some of the features of this molecule in terms of off-target effects. There's very little cytochrome metabolism and little inhibition. There is some inhibition of transporters, no inhibition of the hERG channel that can cause arrhythmias. Brain penetration is quite high, and 94% orally bioavailable means this will be used orally, and toxicity is relatively low. All of this can be done only because of Hande's lab. She's the one that was able to figure out how we can focus on upper motor neurons exclusively, and was able to fluorescently label the upper motor neurons in mice. And then she crossed that mouse with a mouse that had a mutation in one of the genes that's important to ALS, and ended up with mice then that had that mutation as well as fluorescently labeled upper motor neurons. And she's done this with multiple gene mutations, SOD1, TDP43, she's looked at spastin, profilin, And so she has a number of mouse models where you can focus in on the upper motor neurons. And upper motor neurons in mice and humans are really quite similar. And so we feel that using that as a target should help translate our molecules. So with regard to protein aggregation, here's, this is an antibody that can detect the protein aggregation. In the healthy animals, there's very little aggregation, but the ones that have the mutation in SOD1, there's a lot of protein aggregation. If you now take these mice and treat them with 20 mg per kg of compound 9, I'm sorry, of NU9, AKV9, there's less, and then 100 mg per kg, even less. The mitochondria, very important organelle. This is where energy is produced, and mitochondria that are diseased then lose the integrity of their inner membrane. You can see in this top electron micrograph of diseased mitochondria. Inner membrane is broken and the insides don't look very good. But after 100 mg per kg per day of compound, it can be repaired, improves the integrity of the mitochondrial inner membrane. And it brings it back actually to the control, the wild type levels. So here's some data on that. This is percent of healthy mitochondria. This is the wild type mitochondria. This is the mutant SOD1, and now treatment with 100 mg per kg of NU9 brings it back just about to control level. This is SOD1 mutation, same thing in TDP43 pathology, wild type mutant and AKV9 treatment. Endoplasmic reticulum is where the proteins are synthesized. So when that disintegrates, you're going to have problems synthesizing proteins, protein misfolding, and a lot of ER stress. And this ER stress then is what the upper motor neurons, both in patients and in mouse models, experiences. AKV9 repairs this disintegrated ER. Again, here's what a healthy ER looks like. Here's one after mutation, and now treatment with NU9. You can see it looks pretty much like the control. And again, if you look at the data with the control, the mutant, and NU9 treatment, this is SOD1, same with TDP43 pathology. NU9 brings it up very close to the wild type level. The cell body in the brain needs to then extend the communication to the spinal cord through the axons, and the apical dendrites are critical then for this to collect the information so that it then can be transmitted by the axons from the brain to the spinal cord. And if you look at the difference between a healthy apical dendrite and an unhealthy one, there's lots of vacuoles here in the unhealthy ones. And so if we look at the percent of vacuoles of the healthy apical dendrite, pretty low level of vacuoles. But the ones that come from the mutant SOD1, much higher number of vacuoles. Treatment with 20 mg per kg and then 100 mg per kg brings the number of vacuoles down to the control level. Same with TDP43. So again, this is the wild type, the mutant TDP43, and treatment with 100 mg per kg. It's quite low. The health of the upper motor neurons, also you can see, if you look at the healthy neurons in the wild type upper motor neurons, compare that to the mutant upper motor neurons, 20 mg per kg, 100 mg per kg brings the stability of the apical dendrites and back to wild type level. And now the axons, I say, really important. It's got to reach from the cortex all the way down to the spinal cord, and then that message is relayed to muscles. And on the right here, the top, you can see this is what a healthy axon looks like, long with lots of branching, arborization. But the unhealthy ones, it's much shorter axons. And so if we compare now the axon length of the healthy upper motor neurons, this is the axon length for that. This is axon length for the mutant ones. And then this is treatment with 400 nanomolar AKV9. You can see it brings it up at least to the level of the wild type. If you now do the same experiment with Riluzole and Edaravone, those are the two approved drugs. Here's Riluzole, the axon length doesn't compare to AKV9, and Edaravone, same thing. But what was really remarkable was that if you now combine these and combine AKV9 with Riluzole, it's way up higher than either Riluzole itself or AKV9 itself. The combination is much higher. Same with AKV9 and Edaravone. The axon is much longer than either Edaravone alone or AKV9 alone. And so we would hope to test whether the patients then can use these in combination as additional benefit. The mechanism of actions for Riluzole and Edaravone are different from AKV9, and so there should be an additive or maybe even synergistic effect. And so, again, if we look at the actual micrographs of the axons, this is the diseased axon. This is treated with NU9, very long axons. Riluzole, not so long. Edaravone, not so long. But if you now combine AKV9 with Riluzole, very long axons, branching, arborization, and again with AKV9 and Edaravone, very long axons. While all these studies are going on, and the studies, actually we initiate studies at P60 when symptoms actually appear in the animals. So this would be sort of a real life use. You wait till symptoms appear, and then it's run for another 60 days. Body weight is not lost. It's lost compared to the healthy animals, but using NU9 does not have an effect on the body weight. With regard to behavioral effect, if you take a mouse and have it grip a wire mesh and then invert the wire mesh so the mouse has to hold on to measure grip strength, you find that by around P95, they start losing their grip. They fall off sooner and sooner. But treatment with either with NU9, either in the mutant SOD1 or the mutant TDP43, they're able to retain their grip. But as I say, the mutation animals will lose the grip in both cases. As I said, we got orphan drug designation. That was approved, I think, in March or April. And so we have 7 years of market exclusivity after approval. Now, if we just compare, this is a very busy slide, but just to compare AKV9, that's the top line, and what it does, these are all the different attributes of it in terms of protein aggregation, mechanism-based therapy, targets for upper motor neurons, improves mitochondria, improves the upper motor neurons in SOD1 and TDP43. None of the others, either the approved ones or the ones in clinical trials, show that diversity of effects. They are typically focused in a particular mechanism of action rather than multiple mechanisms of action. What's really missing are biomarkers. It would be great if we could identify these problems before they occur. But the biomarkers aren't really at that level yet. However, two of the science advisory board members for Akaba are working on that. Both Hande, Dr. Ozdinler, and Dr. Mitsumoto are working on biomarkers so that we can detect these upper motor neuron problems in patients, and then we're able to have more personalized medicine and know which patients really would respond to AKV9. Just to give you a quick rundown of other projects that we hope to in-license, we haven't yet, but we hope to, using AKV9 for Alzheimer's disease. We've already done a study, a very small study, not a powered study, but we've shown of the looking for memory of objects and their location. All the untreated mice couldn't remember. 3 out of the 4 treated mice were able to remember the object location. We also have a program in inhibitors of neuronal nitric oxide synthase for melanoma. And it appears these small molecules, unlike checkpoint inhibitors, which are antibodies, but these are small molecules, seem to affect this PD-L1 pathway. And so we are looking further in pharmacokinetics and toxicology with that. Inhibitors of ornithine aminotransferase, we've been able to show even at 0.1 mg per kg, inhibits the growth of hepatocellular carcinoma. Inhibitors of GABA aminotransferase, we've actually licensed to other companies, but now we also have an indication in licensed other companies for epilepsy. We have also indication in neuropathic pain, but remarkably without addiction and without sedation or ataxia. Inhibitors of neuronal nitric oxide synthase are also useful in neurodegeneration. We have models in cerebral palsy and Alzheimer's disease that we're working on. So anybody out there that would like to join the fight against neurodegeneration and ALS and related diseases, we are starting to raise funds now. 10 million would be a nice number to get into phase 2. So if you are interested, contact Olga at our company, olga@akabatx.com, and we'd like to talk to you about that. So all the study really, as I say, came from basic science in my lab, in my colleague Rick Morimoto. Hande is an amazing, remarkable scientist. And Northwestern has a lot of ability to move molecules along. University of Pittsburgh helped in some of the very early toxicology studies, and we're funded by a number of organizations to help move this, the basic science along. So that's what I have. And I'd be happy to answer any questions. Well.
All right. Well, thank you. Thank you so much for telling us about this very exciting program. It's pretty amazing, all the effects that you're showing on, neurons in vivo even. So I have the same question as Stephen Parent in the audience. What is the molecular target of AKB, and do you know how it prevents protein aggregation?
Not yet. So of those 3 molecules I said that we identified and optimized from the screen, we did identify the target for one of them. Unfortunately, it wasn't AKB9 because we thought this other one was going to be the one we would go with. And we've identified 1433, 2 of the isoforms as the target. And those 2 isoforms are the ones that have issues in ALS. And so, unfortunately, AKB9 is not competitive with that. So it's a different target, but we're working on that right now to see what the target is.
Yeah. So Dennis, if I may chime in. So I think for the drug discovery, people are looking for one target, right? And I think, remember the table that Dr. Silverman has shown. So there are many compounds that are focusing on one target, one mechanism, but those are the compounds that usually fail in clinical trials because the disease is so heterogeneous that you may be improving the health of a subset of patients, but then that never reaches the statistical significance. So it may not be that AKB9 has just, one enzyme or one protein or one thing. And it may well be that it actually integrates with the membranes and helps the integrity of the membrane, for example, the inner mitochondrial membrane, endoplasmic reticulum membranes, and so forth. And those membrane dynamics also contribute to disease pathology, right? And especially they play a role in lipid homeostasis, cytoarchitectural dynamics, and so forth. So I don't think that we should be looking at just one target. I think as Dr. Silverman has mentioned, those 4 points, right? The targets are the mechanism. So the targets is improving mitochondrial health, improving protein aggregation, improving cytoarchitectural integrity, improving axon length and branching. So those are all targets that we want to achieve. It may not be just one molecule that we're talking about.
Yeah, absolutely. There's a comment in the chat from Robert Hubbard saying that he loves going with functional polypharmacology and drug discovery. And I think we're seeing a resurgence of interest in, phenotypic screening, exactly to address the points that you just mentioned. It's kind of scientifically interesting because you can speculate that there are 2 ways you could achieve that polypharmacology. You could either be, targeting multiple related targets, like, a multi-targeted kinase inhibitor, for example, or maybe you're specifically targeting a protein or target that just has multiple roles, which, 1433 obviously has many, it's an adapter protein, so it could be involved in a lot of things. And I just kind of wonder if you've sort of narrowed this down for AKB9. Is it, something in the, ubiquitin processing family or something like that? Or is it targeting maybe like an E2 or something with a lot of functions?
Mm-hmm. Well, yeah. So I think we have to wait till we see what happens with AKB9 before we speculate on what it's doing.
Yeah, makes sense.
Somebody in the chats asked what the in vitro potency of it is. I think it was 280 nanomolar in the protein aggregation model.
Yeah, we have another question asking about the mechanism of action for riluzole. Does riluzole have polypharmacology also?
Yeah. So I can
Go ahead.
addressing that. So so there are 2 FDA approved drugs for ALS, right? One of them is riluzole, the other one is edaravone. So riluzole actually acts not only the neuron, but also to the astrocytes and non-neuronal cells, and it helps reduce astrogliosis. And astrocytes normally help neurons by removing the glutamate so that there is no excess glutamate, so that there is no hyperactivity. Okay, but then when the astrocytes also become diseased due to SOD1, they fail to remove glutamate, and then the neurons show hyperexcitability. So the mode of action for riluzole is actually on reducing astrogliosis and reducing glutamate toxicity. So that's why I think when we add AKB9 on riluzole together, and that's also, it's to the value of the pharmacology, or what do you call polypharmacology, functional polypharmacology, right? So if you talk about the functions, and I think that's why when you add NU9 together with a drug that works on a different mechanism, different function, we may have additive effects. And edaravone is a pre-radical scavenger. So they remove the free radicals. And again, this is a different mechanism than AKB9. So then I think that's the reason when we add them together, there may be an additive effect. And I think we need to focus our attention more to the functional aspects of the drug discovery.
Yeah, absolutely. And the synergy and the tolerability of the drug is really interesting. That's a very high NOAEL in dog, so very encouraging. One question we have from the audience from Ephemios Gabri is, thanks for the great talk. Could you please comment on the challenges of optimizing the structure of such a small drug molecule?
Yeah, there are a lot of challenges. I have some really outstanding chemists that work with me. And I wish I had time to go through the whole story, because the hit that came from that study didn't look a whole lot like this molecule. In fact, the two rings were connected to one another. And it really, that molecule looked really outstanding until we put it into an ALS mouse model. And this was before I met Dr. Ozdinler. Otherwise, I wouldn't have cared what happened in an ALS mouse model, but it had no effect in the mouse model. And so one of my postdocs thought that maybe we need to have more flexibility. And so she started to add linkers between the two rings, and eventually came on this linker that became U9. And so that made a difference. And then that was active in the ALS mouse model, but it doesn't matter if it's active in that or not. It also now shown to be active in stabilizing upper motor neurons.
Interesting. So we have a follow-up question related to that from Abhinav. He says, what were the molecules that were rejected? Presumably, you mentioned you had a couple hits. Why did you reject the other molecules?
One of them, the oral bioavailability was about 25 or 30%, I think, instead of 94% with this. And the brain penetration wasn't as high as this molecule. And so we thought we'd go with the pharmacokinetic properties of this one.
Do you think those two molecules have the same mechanism of action?
The two other ones? Yeah, we haven't done that. Oh no, they do have, that's right, because they are related. They both have a perazolone core. And so when we did that study, we used both of those molecules in a competition, and both of them were competitive with the photo affinity group that we put onto the molecule. And so those two do have the same mechanism, but this one, as I say, wasn't competitive. So it's a different mechanism.
Interesting. All right, we have a question from Chris Dockendorf. He says, hi, Rick and Hande, very nice activity with the simple molecule and very nice ALS models. Not sure if you can comment on the PK. Is it mostly excreted via the kidneys? Can a 1,3-diketone be glucuronidated?
So we do see a little bit of glucuronidation. There's virtually no phase 1 metabolism, that we've seen from cytochrome P450. The only metabolism, there were, I think, 5 small metabolites that we got out of it. One was glucuronidation.
Yeah, the 1,3-diketone structure also stood out to me. There's this huge push right now to look for lysine targeted therapeutics. Do you think the diketone interacts with lysines?
Well, actually, it's a misnomer for that molecule. It really isn't a 1,3-diketone. The enol form is highly favored. So it's actually hydroxy alpha beta unsaturated ketone. And so that is actually nucleophilic. And so it's possible that it could nucleophilically attack some electrophile, maybe a disulfide or whatever. But we haven't yet looked at that chemistry with it.
Interesting. Okay, let's see. Looks like there are no other it looks like there are no other questions from the audience. So yeah, thank you both so much for coming. Yeah, really fascinating molecule, and wish you both the best of luck in your fundraising and recruitment of your CEO.
Great. Thanks so much, Dennis, for inviting us.
Yes, thank you so much for the invitation.
Yeah.
Thanks everybody for joining us. Bye-bye.
Yep, thanks everybody for joining. And yeah, all right, take care.